Good afternoon, everyone. I welcome you all to the innovation session. Dr. Rohit Shetty will be soon joining us because he's in between sessions. I'll be moderating this particular session. I'm joined by my co-moderator and panelist, Dr. Purna Chandra. Is Dr. Vinit Joshi here, please? Is Dr. Sheetal Brar here? No. Then I would invite people who are presenting to come and join me on the panel. Prakyat, Dr. Ashish, please. Prakyat Sharma. He's also not there. Prasanna Venkatesh also is not here. Okay, I think we can start with Dr. Prakyat Roop's paper first. So, Dr. Prakyat will be talking to us on revolution in presbyopic correction beyond diffractive dichotomy. Dr. Prakyat, please. Can you load Dr. Prakyat's presentation, please?
Mic is on. Okay, thank you. So, good afternoon to the audiences and the panelists on the stage. I am Dr. Prakyat Roop and I am a practicing cataract, cornea and refractive surgery specialist at Roop Netralaya in Meerut. I have a private practice and I have a keen interest in optics, so that is how I stumbled upon this idea of refractive, diffractive technologies and innovations in that, and have been associated with a lot of such research in the past. So I'll be speaking about the refractive technology and the refractive diffractive technology and how they are different, the two presbyopia correction optics that we have, the dichotomy that is there in between them. What is the edgeless phase matching technology, which is a completely different way of looking at the optics and using it for presbyopia correction. And then the clinical results with an IOL, which is the Naturo panfocal IOL, that utilizes this edgeless phase matching technology. The sources of information in my presentation, which I have not highlighted specifically, are from these four sources, that is the US FDA data, the company published data about IOLs, a study that I undertook in collaboration with Lambda X, which is a metrological analysis company in Belgium, and then independent studies done at IIT Delhi and at the Diffractive Optics Group at the University of Valencia in Spain, which is a world leader in terms of diffractive IOL optics. So if we look at refractive optics, to put it very simply, maybe oversimply, refractive optics involves one zone giving light to one particular focus. And so the pupil aperture controls the performance of a refractive lens, meaning that it depends on the size and the location of the pupil as to what area of the lens is exposed, and that changes the performance of the lens. Spherical aberration changes are basically changes in refractive power of the lens that are tightly in conjunction with the change in radius of curvature. So it is a mathematical correlation between the radius of curvature and the radial distance from the center. If we look at spherical aberration based lenses, which are the current day EDOFs and their power maps, we see that a spherical lens has a same power, while if we look at a negative aspheric lens, you will get a slightly lesser power in the center, slightly, or you can say more power in the center. And if we look at something which has modified asphericity to a large extent, that becomes somewhat like a zonal refractive lens, but with a large blending zone. So what it does is that if you look at a spherical lens, the light from the periphery gets focused in front of the light in the center. But if you look at a negative aspheric lens, then the light from the central rays is focused before the light in the peripheral rays is focused. And this is what we utilize to get an extended depth of focus. Now, while these lenses are very, very rewarding, they do have some limitations. So if we look at aspheric IOLs, then this is a very unique article that I saw on CRS Today Europe, where you can get 10 pearls for using a spherical aberration based lens for your clinical practice, which says that if an optical system has spherical aberration of 0, it gives in nighttime driving a 45 feet advantage in stopping distance at 88 kilometers per hour, meaning that if you have a spherical aberration in your optical system, then your stopping speed or the judgment gets delayed by that point. And if we look at spherical aberrations, they become active only at larger pupils, meaning that they are useful in younger patients mostly. If we look at diffractive optics, then it is a completely different way of using the light to get presbyopia correction, because one focus or one zone can give light to multi-orders. That is, it is not aperture dependent. Any exposed zone can give light to 2 or more foci. There needs to be constructive interference of light, for which the radius of zones is tightly controlled by a formula. And the height of each step determines the amount of light going to each focus. This divides our practice into a dichotomy, where on the one hand we have refractive or spherical aberration based lenses, and on the other hand we have the trifocals, which are diffractive lenses. The dichotomy is that while a trifocal lens will give an excellent range of vision, you will have problems in visual quality due to the loss of contrast and due to the night driving halos, glare, starburst, ghosting of light, etc. Now the reason for this problem lies in the diffractive steps. So this is a patent application filed in 1998, which shows that actually light which is incident at these steps causes a lot of scatter and disturbance, which is the cause of all of our problems with trifocals. It says that light incidence from an off-axis point scatters at the steps, and the scatter increases with increasing angle of incidence, increases with increasing step height. And to solve this problem in 1998, before any diffractive IOL was implanted in the eye, it gave a solution. They said you align the light parallel to the incident light. But that is not possible in the human eye. So what is really the need for steps? The need for steps is necessary to match the phase of light to baseline. In simple terms, it is necessary for the IOL to function properly, and it is the best way to do that. At least that's what we knew. And this has been known to everyone right from the very first restore lens, and the reduction in step heights or step numbers has been utilized as a strategy to decrease the side effects. Currently, the most popular lenses either reduce the step height or reduce the step number to reduce this unaccounted light wastage. Now, why was there or why is there no assessment of this light loss to design features? The reason for that is that when you are seeing an IOL on the optical bench, the light is actually aligned parallel, like they said in that application, the patent application. So basically, you are not able to assess the light loss at all. But in real life, light comes in at a slanting angle, and it undergoes multiple optical phenomena at these edges, which causes all the ghosting and starburst and waxy vision. So the lab results actually don't take into account this light loss. And I tried to solve this problem in multiple ways, for which I applied for multiple patents, got granted multiple patents, but practically could not get a working prototype. The second thing that we should always look at is the defocus and MTF. So while a defocus tells you what is the Snellen visual acuity, an MTF tells you what is the quality of that image or what is the contrast sensitivity. In simple terms, MTF is contrast transmission. If you look at the top and the bottom image, the contrast is reducing from the left to the right. But the top image is deteriorating in quality while the bottom image is not. The bottom image signifies a Snellen vision chart. It is a square wave grating. But if you look at the top image, it signifies your real life situations, where it is a sine wave grating. So, if you look at defocus only and don't look at the MTF, you will be confused. Why would you be confused? Is because in a monofocal lens, the MTF for distance is about 80-90%. The defocus says that the vision should be 6.6. In a trifocal lens, the best performing trifocal lens, the MTF is 40%. And the defocus says that the vision should be 6.6. But there is a significant difference in the image quality. Both are sharp, but both are not equally good in quality. So that is why we must look at the area under the curve. Now the question is, why is a patient happy with a trifocal? The reason is that cataract is even worse. The patient doesn't know that he could actually have a much better vision. Now comes to the rescue this amazing technology which introduces negative kinoform phase matching, which is basically saying that instead of having a base curve with a plus add, we have a base curve with a plus add with a minus add. So we have two different kinoforms that are aligned together, and that completely eliminates the edges. Now I did a study on this myself because I wanted to know if it actually diffracts light or not, and we got a ray tracing model made and it actually made sense. So what happens is that this surface, which is the L max profile, it utilizes the complete optical element to give light to a useful focus while eliminating those edges that cause the waxy vision. And that increases the area under the curvature of the MTF curve. We have done about 500 eyes now, and 170 of them bilaterally. And we see that if you look at the binocular distance and intermediate and near vision, at all distances, it is good or better than 6.9. If you look at, I'll take just 30 seconds more. If you look at the distance corrected vision, it is absolutely phenomenal, comes close to 6.6 in almost all of the patients at all distances. If you see the presbyopia range, that is from 100 centimeter onwards to 33 centimeter, as you come closer, it tends to go close to 6.9, which is still better than N6 at all distances. If we look at the symptoms, then the only visual symptom that the patient complains is halos, which is basically having multiple foci, and that causes the patient some problems in about 1.2% patients. The night driving comfort is excellent. If you compare it to EDOF, it is absolutely comparable. Now, this is a very important slide where what we did is, we did a sub-analysis of 12 patients who were implanted with a trifocal IOL and were not satisfied with it. The main reason of being dissatisfied was because of the severe starbursts. And we implanted the other eye with Naturo, and none of them had any visual side effects in the other eye. So we have been using this lens for CLE a lot in post-LASIK patients, in patients who want some spectacle independence but have a monofocal aspheric lens in the other eye. And the patients are really, really happy with the quality of vision and the range of vision. Thanks a lot.
And talk, Prakyat, and kudos to you that innovation is a difficult thing. And the small things, make the quality of life enhancement in patients, and it's extremely heartening to see that you went along with it and brought the lens. Congratulations to you, and we recognize the extreme hard work that you've done. My question is, when you are considering which patient to implant, what is the talk that you have with them? How to understand and how to choose a lens?
Yes, so basically what I decide is, I ask the patient if they want spectacle independence or not. If they are comfortable with reading glasses, everybody knows, and this is going to stay for some time, that a monofocal aspheric lens that makes your spherical aberration 0 for the optical system is the best in quality. There is no doubt about it, like I said. If they want spectacle independence, then I go for Naturo. The reason is that I have shifted from enhanced monofocals and trifocals to Naturo because it requires less counseling. I don't have to look at pupil sizes. I don't have to look at angle alpha. I get predictable outcomes with complete range of vision. Side effect profile is good. So basically, practice has shifted more to it. And that's why probably I am doing so many more numbers of that lens.
Brilliant. Any comments?
Thank you. It's a brilliant work. I have a slightly different perspective to it. So what's your opinion on using these lenses in patients who have some retinal problem like rod cone dystrophy, retinitis pigmentosa, where macula is absolutely normal, but they do have some dark adaptation issues and
I would not use these lenses in those patients. The reason being that if you, like I said, in a optical system which is completely piston, all of the light is going to the retina. So you have to change the focus by using a spectacle, but for that particular point of regard, all of the light is going to the retina. If your retinal sensitivity itself is reducing and you're splitting the light further, then the quality of vision for the patient is deteriorating significantly. So I would not go in for that lens.
so
Definitely.
How about high myopes?
That's a tricky question. So basically, I would go for my retina specialist's opinion in that regard. And if they say that, the retina is quite thin, the outer bands are not that good, the choroid is very, very thin, it's almost like the sclera is right behind the retina, then we avoid it. Because even the ELP in a high myope is not that great. So overall, the outcome is not going to be very satisfactory.
Thank you.
That's
so
the
much.
case with every premium lens,
Makes a lot of sense. Thank you so much. Yeah, thanks.
Excellent. Dr. Sheetal, any comments on this? Sure. Okay. Any audience questions? No. Okay. So I would welcome Dr. Pooja Kamar, a fellow colleague from Narayan Nethralaya. She is going to be talking about gene therapy for keratoconus. Is it a reality? Dr. Pooja, please.
Thank you. And I would like to thank the entire panel and AIOS for planning this session and giving me a space to talk in that session. So, going to be talking about role of gene therapy in keratoconus. Now, why gene therapy for a keratoconic, like a corneal condition like keratoconus, which is a progressive disorder? Over the years, starting from 2010, our group has worked on role of lysyl oxidase in keratoconus, and what they have proven is that lysyl oxidase is lower in patients with keratoconus, and with increasing grades of keratoconus, the lysyl oxidase does reduce. And also, if the lysyl oxidase is lower to begin with, and if you do a cross-linking for those patients are at higher risk of treatment failure, and they still can progress in spite of doing a cross-linking. And not just that, patients who have developed post-refractive surgery ectasia and SMILE, they also had lower lysyl oxidase to begin with, and that's why, like, the topographies and everything were normal, but because the lysyl oxidase was on the lower side, they landed up after surgery developing an ectasia. And we do have seen reports again at places that they have done a transplant, a DALC or a total corneal transplant, and they have also failed at times, suggesting that yes, there is a genetic disorder which we are considering. So we wanted to ask a question that can we explore the role of enhancing the lysyl oxidase expression in the corneal cells and the tissues, and is it safe and provides potential functional benefits when it comes to keratoconus therapy? Now, the ideal gene therapy is going to be something which is safe and non-immunogenic, introduction of genetic material by an able vector into the targeted cells, rendering them capable of repairing, replacing, or overpowering the functions of defective or non-functional genes for therapeutic purpose. So our hospital has developed this vector production facility, which is one of the first in India in an eye hospital, where we can produce our own vectors, and what the vectors we have chosen to produce is the recombinant AAV vectors. Now, why an AAV vector? Because those are known to have efficient delivery of genes of both dividing and non-dividing target cells. They are known to give high levels of gene expression. There is an absence of viral genes for causing an undesirable immune response, and it helps us to have a successful in vivo administration to patients without significant toxicity and excessive immune reaction. So that's why an AAV-based vector gene therapy. Now, this study is a 3-step. We have reached the third step, and soon we are going to start the clinical trials as we've gotten the approvals now. So the first step was to do injection of this vectors, LOX vectors in one eye and the controls in the other eye in an ex vivo cornea model. And once it was injected inside the cornea, post 6 weeks, we studied the expressions of these genes like lysyl oxidase, collagen 1, 4, and MMP9. Now what we saw was that when we injected the lysyl oxidase, the levels, not only the expression increased for the lysyl oxidase, but you can also see that the collagen has started resynthesizing and it increases the levels of collagen 1 and 4. But what advantage we saw was that it was not only increasing the collagen expression, but it was also reducing the inflammation. Now inflammation and keratoconus goes hand in hand. Whenever you have inflammation, there is a higher risk of progression. So this was giving us a dual benefit, that it was not only enhancing the expression of lysyl oxidase and collagen, but reducing the inflammation as well. The second step, we wanted to see how does it behave in a donor eye, in a human eye. So we took the human donor lenticules from the SMILE procedure, which were not discarded, but they were used for the same study. And one part of the lenticule, one eye of the lenticule received the lysyl oxidase and the other received the control. And again, the similar results what we saw in the ex vivo cornea model were replicated here as well. Now the third step was to see that what actually happens, what we are seeing in the gene expression analysis, the quantification. Do we see it qualitatively as well, and are we able to, like, what is it like, because the collagen levels are increasing, is the strength of the cornea increasing? So this was done in whole eye globe from a donor which were not suitable for transplantation. We created a pocket, intrastromal pocket, which can be used for camera implants like an intracorneal ring segment. And again, one eye was used to inject the lysyl oxidase vectors and the other was the GFP. And you can just clearly see in these two images, this is the control image and this is the lysyl oxidase image. The spacing, the white spacing here, what you can see is nothing but the green bundles are the collagen bundles and the white spacing besides them is the gapping. But whenever you inject these vectors, you can see the spacing between them is reducing, and the blue color effect, what you can see, the collagen bundles are increasing in phase. So it's not just the quantitative effect what we see, but the qualitative effect as well. Now atomic force energy is something which can be used to study the structure and mechanical properties, and it can be used to measure the stiffness of the thing. So again, you can see here that the eye which received the lysyl oxidase, the elasticity does increase, in saying that the Young's modulus and the stiffness of the cornea is increasing. And this was seen not only in the cornea, goat corneas, but in human donor globes as well. So yes, gene therapy in cornea does have novel insights. It helps in developing non-immunogenic vector, and it also looks at the patient safety. And what is the best advantage when it comes to cornea is we do have imaging modalities which allows us to see the structural changes and the biomechanical changes and whether it's really affecting it or not. So lysyl oxidase gene therapy may be a potential treatment modality for keratoconic patients in future, where transplants are not an option or where cross-linkings have failed. Thank you.
Terrific work, Dr. Pooja. Dr. Sheetal, any comments on this?
This therapy currently you are utilizing for keratoconus patients.
We will be doing it for keratoconus. My colleague Dr. Poorna and Dr. Thiru, they work on the retinal front. So I'm working on the cornea front for keratoconus.
So how long does it take for to see the changes in the confocal or the imaging that
so
you saw the bundles? That
so
the
this
spacing
was
was
done
reducing.
at 30 days post-treatment.
Okay.
But so as I mentioned, we have received the clinical trial approval and we may start anytime between April or maybe next quarter. And I think that time we will have to see effect at day 1 month, 3 months, and so on.
So these were
This
porcine
was,
cornea?
this was on human corneal globes,
Human
the
corneal
ones, yeah,
globes, okay.
cadaveric eyes.
So
And
if
the
I'm getting the
lenticules.
future correctly, if I'm envisioning it correctly, what we are going to do is basically we're going to inject this, we're going to correct the keratoconus, and then we are going to correct the refractive error.
I
Or
will
make the shape
not
better.
say that you're going to correct the keratoconus. I will say that you are using it in patients who basically are trying to avoid a transplant for these patients. And as I mentioned, when the lysyl oxidase or collagen levels are on the lower side, your cross-linking effect may also fail. So this would be an injection, like when you inject and increase those levels. So even if tomorrow you want to do a cross-linking or a transplant, it may work better. So the aim is not to reverse them back to normal, but to stabilize them.
So
And
early keratoconus cases it can be done.
probably moderate to high keratoconus cases.
Okay. So in early cases, cross-linking, the regular conventional.
Conventional cross-linking or a medical
treatment,
cross-linking,
yeah.
what we have been working
And
on.
for the moderate and high gene
Yes,
therapy
yes.
with or without cross-linking?
Yes.
What she's looking at is that, you know, in rats we know that it is increasing collagen. Initially, because we'll have to demonstrate safety first, right? So initially we are going to take advanced cases, do this, look at how they do, and efficiency will come later. So too early to say that we will be preventing keratoconus, but we'll be definitely managing them better in the early part. Once the safety is established, efficacy will come along, and you know that will give us more knowledge to really say that we can use drops or injections to the cornea rather than replace
Do
it.
you also see its applications in refractive surgery?
So, yes, if you have a poster, so what we are working on, as you are well aware with it, we do work on various kits
Yeah.
which can measure the lysyl oxidase levels, and we want to do that because one of the factors what we have seen is post-refractive surgery ectasias is because of low lysyl oxidase and collagen. So tomorrow if you feel that, you know, everything is healthy but the patients have lower levels, when we have, which will be maybe few years down the line, when we have this in a drop format, probably treat them with drops, enhance the expression of lysyl oxidase collagen, and maybe 6 months or 1 year later do a refractive surgery. But
Even if
that's
we
a very long
I know,
future
but
dream.
even if lysyl oxidase, that kit is not yet available, and this is available, in early cases of ectasia, do you think it can also help?
Yes, definitely.
How it works in
Definitely,
keratoconus,
yes,
right?
definitely.
Pooja, yeah, I mean,
Yes,
I
sir.
am a retina specialist, but I have a slight inclination for basic science research. So, my question is, for me, diseases like keratoconus, even a lot of retinal diseases, the pathology is kind of multifactorial. So, are we confident on addressing this one part and then taking care of the keratoconus overall? What's the
No,
comment
as
on
you
that?
said, it is a multifactorial disease and there are multiple factors. So this therapy would probably be working well where you know that there's a genetic defect, where you know the lysyl oxidase are on the lower side. But if I know that the lysyl oxidase levels are optimal, but it's the inflammation which is leading to progression of the disease, I would address that part of it. So once we have this in hand, I think we have to have an algorithm of where to choose a gene therapy and where not to choose a gene therapy for these patients. So
And
it's
we
not
have-
going to be for everyone.
Yeah. And we have already identified the predominant gene which is involved here. Is that
Yes,
right?
so we have identified predominant genes here, and that's why we are looking at the enhancing the lysyl oxidase expression.
Thank you.
Thank you.
One thing, Ashish, is when we talk about genes, they have a lot of crosstalk. One crosstalk which I feel has a huge advantage, not just in cornea, in everywhere else is, when we altered this gene, it drastically reduced the inflammatory pathway. So that inflammation for some reasons was knocked off. For example, the MMP9, and she must have shown. So what really happens is it gives you a collateral thought process that when you create a gene therapy, say for ARMD, like what Thiru and the people working is, you might actually knock off a pathway which is not the gene itself, but the molecules which is inflammatory, which can offset the thing. So this is something which we learned very new. So tomorrow, the gene therapy could be an effect for Steven Johnson when cornea is getting lysis, or a scleral melt, where you're looking not just creating a collagen there, you're offsetting a pathway. So that is a completely new aspect which probably we don't know.
Speaker is Dr. Prasanna Venkatesh, and he'll be talking to us about the novel 3D effective holographic ophthalmic AI metaverse with augmented reality. Dr. Prasanna, please.
Yeah. Thank you, Rohit Shetty sir, for giving me this opportunity on this innovation session. So for the next few minutes, I'll talk 4 components of the innovations that we have done. One is the teaching pedagogical one, another one is the clinical diagnostics, surgical simulations, and then the AI. So things which was once thought to be a science fiction, all of us would have seen this movie or movies resembling holograms, which was once thought to be science fiction, is now no more science fiction, it's a science-based reality. So we started slow and steady with the LiDAR technology that is available in the smartphones, and this is the QR code for Android and the iOS that have innovated respectively. So the models were actually created with a template, a 3-dimensional template, and in the smartphone it was given for the user. So you can see that the right hand side, the video is actually synced with the left hand side. So the pedagogue or the teacher who's actually teaching the students onto orientation. So we wanted to first start with accelerated learning or pedagogy. So on day one, can we show how the lens or the fundus or the anterior segment structures really look? So you can see that the videos are actually synced with each other. The right hand side is actually synced with the left hand side, and you can see the phone actually navigates. Yeah, the phone actually navigates as a three-dimensional spatial tool, and you can go ahead and see the fundus as well as the real fundus images were given, and also you can see the angle structures were used, a photoreal visual reconstruction for the angle as well. And this is again another video of the phone where we did a screen recording. You can see even a beautiful angle structure which mimics as real as possible for an AIOC counseling or for teaching the structures as an SLT or an ALT for that matter. And we navigated towards to the fundus as well. You can see this is a normal fundus. Now we have an NTG patient where there is a splinter hemorrhage, and also the color fundus or the autofluorescence can also be collected. And then you can see we have moved towards an RNFL defect. So even you can use this for retinal pathology. So the idea was teaching pedagogy. So we started with making an atlas of fundus, atlas of angle structures. Can we have something to have a pedagogical augmented reality teaching tool in the smartphone? So that was how we started, and we validated it and published it in the European Journal of Ophthalmology 2023, that is last year. And then also there was a commentary on the same in the IJO 2022. Then we wanted to push ourselves further in can we have a subtractive module for a virtual dissection. So ophthalmology and neuro-ophthalmology structures, we tend to not really get to do dissection of structures that we wanted to. So we started giving buttons here, and the user can actually choose buttons. There were no app available that can actually subtract structures and then go in and see the structures. So we sat with our anatomy and we read the anatomy, reconstructed it whole in 3D, and then you can see the user can choose and use subtractive learning, and that with augmented reality was possible for neuro-ophthalmology brain as well as for the eyeball models was used. And then pushing ourselves further, can we use some simulative modules for smartphone? So in this on the left hand side you can see, so the models have been loaded, and now I'm moving my smartphone towards the pupil. So this is like a direct ophthalmoscopy. So you can talk however you want on direct ophthalmoscopy, but none like actually showing them this is how you do it. So you go in, you come back. So when you do it multiple times for field workers or for your neophytes or your postgraduates who are starting their curriculum, this really makes a lot of difference. And also for e-counseling on the right hand side you can see, we have seen that showing their patients their own images in a smartphone, I think it makes a whole lot of difference. And we are trying to move into EMR as well with their own case sheets coded to them at the end of the day. So then we pushed into the headsets, that is the ophthalmic diagnostics. So the same models that we had created for smartphone learning, with this headset, the HoloLens that we got, so you can see that the models were actually embedded into the meshwork, and the touch with a haptic sensation, a pseudo 4D or a three-dimensional orientation, where the same examination of the patient, eye being a three-dimensional organ, can be examined 3D itself. So you can see the same splinter hemorrhage patient case sheet. So now we can examine it in 3D, the angle structures can be extrapolated and examined, and then you can see the RNFL defect here, which is being examined in color as well as in autofluorescence and infrared. So we published it in the August issue 2022 in the Indian Journal of Ophthalmology, and now we are pushing ourselves further to incorporating OCT also into it. So when a patient walks into the hospital, you get it into the software under 60 seconds with no instantaneous chair time, you get the holograms of the OCT extrapolated into it. And I think eye as a three-dimensional organ has to be examined in the 3D with the imaging that we have, multimodal imaging, and before and after treatment you can counsel the patients with it. And this is again another collection of flecked retina and retinitis pigmentosa, and also for low vision rehabilitation counseling. Mostly if you see, I think we are seeing it in neuro. This is making a lot of inbounds because cognitively tend to imagine it, and with subtractive element added into the holograms, it's being a game changer. Moving on to the third part of migrating into the surgical simulations. So this was an idea when I was doing one of my MAGS cases. The stent was superficially implanted. This was a 2022 case of mine where I had to place the second stent first, Then I had to take and reassess the implant that was actually hanging because it was an under-implanted one. I was almost losing it in the phaco wound because that time there were not many teaching modules in 2021 when MIGS were hitting India. So all's well ends well, I was able to recover the stent and then place it well, and the patient is a happy patient. But then I went back to the drawing board, and can we do something for surgical simulations beyond cataract, because cataract, phaco, SACS are something which is well taken care of. So that is when we went back with this particular HoloLens, the three-dimensional one, where we went back to the drawing board and we had the anatomy of the angle already there. You can see that the left-hand side and the right-hand side are synced with each other. On the left-hand side, you can see actually what the user is seeing through his eyes. So we made the MIGS more easy because this is three-dimensional spatial orientation. So every time when you implant the stent correctly or when you do a bang or a KDB, it gives you a visual cue that you are doing it right. So you practice more and then enter into the real field so that you get it right. So we patented this innovation of ours, which is available for HoloLens users as well as for Geo users, and also published the same in IJO. So coming on to the last part was the artificial intelligence, which goes hand in hand with these kind of inventions, where the software application which we innovated, we did a different concept of human intelligence with AI. So we wanted to utilizing human intelligence in AI because AI is ultimately like human. It has a CNS just like the neural network of the humans. It has to undergo a growth phase. So we train the machines by having two human interfaces, having a human annotation to train it, having a human validation to correct it if it's moving in the wrong direction. So each image was sat by a human ophthalmologist, a glaucomatologist, and a retina person, and you can see the amount of depth. Some images were taken 15-20 minutes to train. So at the end, you break the black box. Sometimes in AI, we don't know why it is predicting the way it is predicting. So we wanted to have the diagnosis along with the findings. You can see even an RNFL defect can be predicted, and explainable AI is something that we are moving forward to, and we are pushing ourselves further and further. So now we are working on anterior segment protractors as well for diagnosing where is the trabecular meshwork, the green dot that you can see, then identifying the scleral spur, where is the scleral spur, then getting your protractor there. And though there are many angle protractors, now with the mix that is coming in, when you open up the angle which I'll be showing next, so we need to invent and reinvent ourselves because there is more to understand in this region after you make the procedure. So we are pushing ourselves with UBM, Anterion, ASOCT, and Shemflug imaging. It's a multi-model project that we are doing, and you can see with every training, the results are much better. From 24, 27, 24 is now 22, and now it's as one difference as much as possible. So this is something that is very promising. We are moving to OCT as well. Me being a glaucoma person, I am pushing myself more into glaucoma per se. So how we can diagnose, not only diagnose, but also grade the glaucoma with severe, moderate, as well as differentiate the glaucoma with the anterior segment modules. And this is the study that I was talking about, which is due for publication in IJO. There are 5 to 6 different mixed surgeries. Each angle anatomy is different, and we are working on these segments so that we get the outflow channels the way it is, how the Schlem's canal is opening up in different procedures, be it the excision procedures like a KDB, bang, or a suture gate, or stenting procedures like an eye stent or eye stent inject. So every model has to have its own growth, and there is no end to this learning, my dear friends. I am using human in the loop machine learning, where human goes hand in hand. You can use it for diabetic retinopathy, and you can use it for what not, OCT, and there is no ending to it.
I think you have to sum it up.
Yeah. So that's all there is to, my dear friends. We are going to have an hands-on between the machine and the thing for a better humanity, and I would like to thank once again, and this is my team behind it. Thank you once again, sir, for giving me this opportunity to cover on all the 4 different facets. Thank you.
Start with Ashish. No, comment on this, all the retina people, then Sheet.
I just had one question. So does it show the actual patient's pathology, or let's say they're having macular edema and the actual picture of the patient, or you just have models of different pathology and you just show
This
them?
is not an imaging tool, ma'am. It is a translational tool.
Okay.
So this is not an imaging tool. Yeah.
So I have a question, Dr. Prasanna. How repeatable is it? Like when you try to counsel the retina, is there an eye tracking system? Like first time you see something and you counsel this is there. So when you repeat it, it has to be accurate so that when the minor details
So with the spectralis that has come into the field, sir, there is a Brooks membrane opening for fovea BMO axis. So with that, what I am getting is we are able to get exactly the macula where we want to and the optic disc where we want to. And incorporating the lens star along with it, even the eyeball biometry and this fovea BMO axis, we are able to reconstruct the models as precise as possible. So I am using those technology also into creating these
That
models.
is what you're planning to.
Yeah, we are already doing it, sir, but we are refining it as we speak. Yeah.
So
Thank you.
one thing you could, because you already have a
Yes, sir.
3D visualize, one thing you should really consider doing is incorporating macular perimetry
Okay, sir.
into that thing, because see, if you're adding your OCT images there, you have a fundus images,
Okay, sir.
and if you're able to look at the macular thing. And one thing which is which I feel is very necessary in today's, it was there as a one tool. It used to be, it's particularly Maya. Maya was stand for macular integrated something. See, it was giving you a preferred retinal loci, multiple things was giving. That machine does not exist.
Okay.
Using the same stimulus, you can look at how you can build those macular responses. Something you can incorporate
sir.
in it, which probably will become then a teaching module and also a diagnostic module.
Definitely will work on it, sir.
Ashish?
Yeah.
Prasanna, great talk. So,
Yes, sir.
means I just want to know that where do you see after 5 years means being
Sir,
used?
we have the applications ready. It is just that the products have to be, I mean, there has to be many institutes forthcoming with this 4D holographic simulators, be it Oculus, Geo. If Geo is going to come up with something cheap, our apps are ready to be launched. It is just that we have it in the cloud, we have it in the mesh. So I think we are taking one day at a time. So if the HoloLenses or the Geo lenses are ready, our apps are ready. So it's just that the smartphone is not ready, but the apps are ready, sir. Yeah. Thank you, sir.
I think which is also very important, because with the same thing, we tried initially, but we were unsuccessful then, but I think we then left the project. I think Roop is also interested in this, is to add simulated essay onto this whole model. So that means that you can build a real time, but you know your EDOFs and all these lenses are all the next generation lenticle procedures. So he has some ideas, so you both can look at collaborating this to see how you can simulate that. Adding essay change simulate is a little challenging, because you need a different, you cannot build an adaptive optics onto your camera. It's not so easy. So we did not really succeed. I'm talking about 10 years back. I think Roop can look into this kind of model to build on this. Yeah, but at an idea like what he said, like an idea, you can start working on it, because that's what is commercially needed. Yeah. Like to call upon Dr. Tirubalesh. He's going to speak about macular degeneration, newer targets from drugs to gene therapy.
Thank you, Rohit sir, for this opportunity. Good afternoon, everyone. This is the team that sir had put together with the aim to improve what we are going to do in the future. We know that for AMD and DR, we are giving multiple injections. We want to look at a future where, maybe a single time treatment would be effective, and also develop newer treatments for dry AMD. We are in active collaboration with Dr. Sinha's lab, and our NHP experiments are done at SNEC. So I would like to acknowledge that. So AMD, we all know, is a multifactorial disease, and we know that there is both dry and wet form, and there is no treatment for any of the dry form. There are about 250 to 300 genes which have been actively correlated with dry AMD, but the thing is, if you knock off any of these genes, you cannot produce an animal model. say for example, if I block the ARMS or the APO gene, you cannot produce a drusen in an animal. But in 2018, Dr. Sinha published this work that there is a gene called as CRYB1. This is related to the beta crystallin. Embryologically, beta crystallins are associated with development of the lens. But beta crystallins also have a function where they can modulate RPE function, right? So when he knocked off the beta crystalline gene, what you saw was that there was development of drusen. So presenting to you for the first time an animal model where if you knock off a gene, it can produce AMD-like lesions in the form of drusen. You can see that, after 4 months of the knockout, there is active appearance of the drusens, and this is on the left-hand side. You see a image of the fundus of the mouse where it was not knocked off, and there is absolutely no drusen. Now, the safety of the vector production and all, we were evaluating, and we were looking at animal trials. We did the rodent studies first, like I showed, and we took it up for using this and in primates and chimpanzees. So what we were asked to show first to give the data to the FDA is it safe? So we injected it into the chimpanzees, and we studied doing OCT, and we also did histopathological studies. The OCT did not show any kind of toxic kind of reaction when we gave this drug in the suprachoroidal space. You can see high definition. These are not human OCTs. These are all OCTs of the chimps. And we also did a fluorescein time to time to check if there was any vasculitis kind of reaction, because we were injecting a vector, and we found none of this there. But we also wanted to show that there was integration of the gene therapy product which we have injected, and you can see in the green, it does definitely go to the RPE and it gets integrated into the RPE. So what I'm trying to tell is this particular gene can be used in AMD and reverse the process. What it actually does is that it makes the lysosome more efficient, it makes the cell younger, it makes the cell function like what it was 10 years back, so that the disease progression is now suddenly taken back 10 years before, so that the chance of progression to GA would be grossly reduced. So that is the concept we are working on. We have huge amount of primate data available, and this will probably go into clinical trials very soon. Now we looked at another pathway with Dr. Sinha's lab. What we looked at is there is something called a C-gasting pathway. This is already published. What we saw was that there is a cyclic AMP to GMP conversion enzyme, which is the activated C-gas. This particular thing can activate what is called as the STING pathway. What do we mean by STING? Is stimulator of interferon genes. So what is the role of it? I'll be speaking in the next couple of slides. We know that DR is a disease which is vascular in nature, but with DR studies, we learned a lot that DR is premature senescence, right? So when we looked at the mouse study, we initially looked at only the leakages when we were dealing with anti-VEGF agents. But when we looked at the protein expression, we came to know that in DR also there is a lot of cellular senescence that is happening. Glucose can make the cells age faster. Premature senescence is a feature of diabetes which is already established. And we looked at 3 important factors, which is the P16, P21, and P53. As we age, these are the markers which get more and more produced in the body. So these are definitive markers which are associated with senescence, and that we also saw in DR. So if you don't brand the disease and understand the disease, senescence can also be understood by studying DR, right? So what we did was, we looked at, if in the DR part that there was a lot of inflammation that was happening and that was directly related to the C-gasting pathway. So we had an idea that what if we block selectively the C-gasting pathway in the endothelium of the patients and also in the RPE of the patients. So I will show you, this is the proposed mechanism which got accepted in JCI Insight, where we showed that C-gasting pathway is at the heart of it. The senescence is actually dependent on this particular pathway activation, and this is what causes cellular inflammation. So our idea was, if we knock it off or if we make it defective, what happens to senescence? Now I'll show a slide where, in control patients, you see the C-gas is a lower level, but the moment there is DR, it goes up, right? And the STING is also going up. So both C-gas and STING are live, and these are from the human donor tissues. And you can see that in the control and DR, as it becomes more and more severe, the interferon levels and interferon beta levels are going up, which kind of aligns with the theory that we're talking about. What we did next was, we produced a mouse model where WT is the wild type, STING KO is the knockoff part. What we did was, we knocked off the STING pathway, and we also produced the defective STING pathway. What we observed in the mouse and in the protein in this thing was, PTPK1 and IRF3, these are tank binding kinase and interferon regulator genes. These significantly go down in the knocked off mouse model. What it essentially means is that if you knock off the interferon gene pathway, the mouse actually did not develop diabetes despite giving them streptozotocin and making them more senescent. Now, this particular thing tells us that if I am able to silence or conditionally block the Sting pathway, how it is working in DR, it will definitely also work in AMD. We have Dr. Ashish's talk, and he will be talking about anti-VEGFs. Anti-VEGFs work both in, what was working in DR was also working in AMD. Now, this is associated with senescence. We proved that there was premature senescence in diabetes in all the animal models. So, if we block the senescence-causing genes in AMD as well, it might definitely provide a new target in the coming few years. So, exciting times coming up where we will be having newer and newer modalities to block these kind of senescent disorders. Thank you very much. And I just have one more slide. This particular thing was, it's already in human trial. This particular paper got published in Nature, and these people actually proved our concept by using a biological and blocking the senescent proteins, and they are using it for treatment of AMD currently, and it is in phase 1 and it is showing promising results. So what I'm trying to tell is, if it is already there in human trials, and I think we are not far away from developing this target. Thank you very much.
One thing which I would like to open this with discussion, I think Ashish is also speaking about the biosimilars, and he's pioneered biosimilars in a big way, is we all talk about different injections working on different people, but one thing apart from the gene therapy part is each one of us have a gene tolerance to certain biosimilars. So that means geographically, embryologically, or evolutionary, each one of us have a certain genes which works very well for certain type of biosimilars. I think that is something which we have never studied because it's a very, very costly thing to study. That's the reason, some biologicals work very well in certain set of population with a certain set of genes. And it works completely opposite in some places. And sometimes what works, that works exactly the same thing works on immunomodulators on the ocular surface, including dry eye. So one question is that there is an important pathway called NF-kappa B pathways which you're trying to modulate along with the thing. NF-kappa B is also responsible for the ocular health also. So any gene which knocks it off is also going to compromise the health. So how do you target in such a way that you make it less active and off the action, and suddenly it's like switch on and off mechanism. That's what Dr. Pooja also mentioned. The most important thing about gene therapy is it's like a switch mechanism. You should have a switch, we should on it, and when its work is done, you should off it. In corneal gene therapy, we use doxycycline as a means to use a switch. But what do you use here? Because it can't work forever.
Exactly. What we are doing is that this particular silencing that we're doing, one is it is very localized. So we have developed a vector which is extremely specific to RPE. So if I do a gene therapy in the eye, if I put this particular thing in the suprachoroidal space, it is going to bind to RPE alone. It does not have affinity for any other cell. So although I am silencing inflammation, I am silencing inflammation at the RPE level. So it diverts that particular.
Ashish?
Thank you, Thiru, and excellent work, and congratulations to the entire team of Rohit and Thiru. So Thiru, I have one question, means if I have understood it right, the space that you have used is suprachoroidal space, isn't it?
Yes.
So we have a lot of therapies now, we have reached to a level that you have phase 3 trials in, with intravitreal, suprachoroidal and subretinal. So as of now, what works the best is subretinal. So means, did you try it, or if not, then why did you not try it?
Yeah, that's an excellent question. Thank you very much. So Jean Bennett, who is the inventor of the treatment for subretinal, she told us that subretinal works the best. But I showed you animal trials, and I showed you the chimp data. So we have compared both subretinal injection and suprachoroidal injection. And what we found was the amount of expression that was there with subretinal injections, we were able to get it in the chimps by giving a suprachoroidal injection. Now this achieves two things. Doing a subretinal injection is such a complicated surgery. If this comes into vogue, how many people will be able to do it, right? So that was at our heart of when we were looking at this innovation, that we wanted to make this an office procedure. And suprachoroidal injection is as easy as giving an intravitreal injection. And we have developed a patented needle also, which is going to be about 30 gauge. And it is going to be just like an office procedure. The patient walks in, takes the gene therapy without undergoing any major surgery, no risk of detachment. And if it works, this probably is going to revolutionize how we are going to see the future treatment of AMD and DR.
So this leads to the last query. So for example, you did test subretinal and suprachoroidal. Did you try intravitreal? I know that's a route which causes a lot of inflammation, but that's the easiest if we are looking at the ease.
That's again a brilliant question, because we know that vitreous has a lot of APCs, antigen presenting cells, right? So although the gene probably we wanted to silence here will get knocked off by the APCs because they chew on them and they produce antigen readiness within the system. So if it kind of causes that kind of a reaction is what was our concern. We did give intravitreal. With intravitreal, we found that there was some amount of inflammation. So that's why we stayed away from it. And like you said, it becomes even more safer if we are able to kind of divert that kind of APC regulations on that. But then it's a good idea to actually look into.
Thank you. Excellent.
I just have one question. Who will be the patients who will benefit maximum from this? Is it the early stage of ARMD or late, or you can use it for prophylaxis also?
Again, that's a brilliant question, because initially this treatment we are going to use in advanced cases. Like I said, any new treatment, before we start giving them to the original target population, we have to show, although we have shown in chimps that there is no inflammation, there are no side effects, when we give in human patients, we are going to look at them for quite some years, isn't it? So we are going to first give in advanced cases and prove that there is no reaction that is setting in. Then we are going to use in GA cases and show that there is stoppage of progression after doing a follow-up for up to 2 years. And then we will apply for what is called as giving this particular treatment in early cases, like patients who are having moderate AMD where there are confluent drusen, where we know that the risk of progression is very high. That is my original target population. So if we stop the confluent drusens, you are preventing a CNVM at some point of time. You are taking them back 10 years before. We are not saying they are never going to develop AMD, but the development of AMD is going to dial back to 10 years.
Thank you.
Thank you.
So thank you so much, Chiru. Now I would like to invite Dr. Ashish Sharma to deliver his talk on why innovation should travel from A to Z. Over to you, Dr. Ashish.
Is this the one?
So I request AV team to please display slides.
Yeah. So good afternoon, ladies and gentlemen, and thank you, Rohit, for having me here. Rohit sent me a message at 11 o'clock in the evening, or rather night, that Ashish, I want you to speak in this session which I'm organizing, and then I said, okay, give me a little time to think overnight that what topic I should be speaking. He asked, can you give me a topic? So the topic is why an innovation should travel from A to Z, and I'm so glad to see Prakyat. I am so glad to see another colleague of ours, who traveled from A to Z. So before I proceed further, these are my disclosures, and I do have proprietary and financial disclosures here to disclose with MI Redkin YNC. So what do I mean by A? I mean A means when a thought starts in terms of innovation in your mind. And what is Z? Z is where you have your product commercially available and you see in hands of people. And that is making some difference. Now the journey from A to Z is something, which you need to travel. And when Rohit asked me that, you should talk about it. So whether I am worthy of talking about it or not means I'm not sure, but probably we have had traveled from A to Z in some of the products that we could thought about it and we could commercialize it. And our first product was MI Retcam, which has started its journey in 2013 as A, and it's reached Z in 2015. And at present, on podium I say, it is present in 67 countries with 16 scientific papers published, and we have received patent last year. This is another device that we again traveled from A to Z, starting A in 2018 and reaching to Z in 2020. I would not take credit for this. I had a brilliant student of mine, secondary DNB student, who came to me, Nilesh. He said, sir, you know, I want to develop a device which can really capture the angle images, because as of now we verbally say, but can we do this? I had a straight question to Nilesh, are you ready to travel from A to Z? Because I do not really believe traveling in between. Nilesh said, yes, sir, I am ready for that. And this is the picture on 14 September 2020 when we were packing our first commercial device to dispatch. And image which was captured with this device was a part of official journal of World Glaucoma Association, Journal of Glaucoma. So that was the kind of testament for the quality. So we have, as of now, 3 products which have traveled from A to Z. Now, the question is that why a product and innovation should travel from A to Z? What you need, if you are not a saint, then you need these 3 things: name, fame, and money. That's what predominantly every innovator is trying to travel along, except saints who do not, worry about the last part, but majority of them, they do. Now, what happens with predominantly majority of the time when I speak to students, postgraduates, early practitioners, P is a stuck point. P is a point which gives happiness, but it makes you stuck at that point because the journey from P to Z is slightly complicated. What I mean by P is presentation. You have a concept, you present it in India, you present it overseas, you get applause. You are happy. You sit back and relax. Second, your idea got published in a very good journal, high impact journal. You are happy seeing the journal being cited few times. Again, you sit back and relax. Then third comes when you are quite close, prototype. Prototype is somewhere, now things have started working. So a lot of people I see, they are happy with Jugaad. But when Nilesh came, and any student who talks about innovation, I have one point to say to them, that people have money. People do not have time to invest. So if you bring with a product, people will use it, pay for it. But if you bring a jugaad, less likely that somebody would be using it. So this was our first publication, 2016, after we filed a patent. You see a brilliant citation. It is cited by 89 co-authors across the globe, because that was something which was new, where we were able to image peripheral retina up to the ora serrata and pars plana as well. So that excited people. We could have been just excited that time and then stopped working and see, this is the journey which has gone from the drawing board till prototypes. And then prototype was something which was published in Euretina. We had these brilliant images coming from the device which was at the prototype stage. So these are, I'm talking about, P. These were P's which were giving us happiness. But then we were quite close to Z when we had our commercial model ready by the designer. But even once you have the design ready by the designer, from that to Z accomplishment, which is a final product of you, sometimes there are changes. So we had ergonomic testing, we have a lot of other testings in various hands, various phones. Then final Z was accomplished and the design was slightly different. So as I said, again another P was there, even the product was not ready. We were everywhere. We were at academy, courses, World Ophthalmology Congresses. We could have been stuck at P again. But what happens, as I was mentioning in earlier slide also, from A to Z, if you are stuck at P, what you get? Name and fame. But what about money? So if you are not saint, what money can do? money inspires you to innovate further. So you need to see that money, and that money comes only if you travel till Z. And what money does, which it has done with me as well, when during the COVID time, when our salaries were deducted, we were not paid completely. It became the secondary source of income for as a person to me. So we really talk about in our financial sessions, nowadays in ophthalmology conferences also, that you should have some second source of income keep going. So this could work in that way as well. Another point is, if you are stuck with publication and presentation, you can be forgotten very easily. And what traveling to Z does is, it creates an immortality of you. You live really long once you travel till Z. Now, another question is, can every innovation travel from A to Z? No. We are stuck with 3 innovations, not even at P. We are stuck at multiple levels. Means RIGS is my dream device. When people are, coming to a diabetologist clinic, they wear a glass, then glass takes a picture. Every 6 months, patient wear a glass. Then the images are transferred to a Bluetooth on your phone. Then AI works, and then you get an answer. So until unless home care comes, you will really not get any solution for diabetic retinopathy blindness. I'm quite firm about it. A lot of work has been happening, so we are stuck there. With the smartphone itself, we are trying to image where we can image even up to the pupil in one go. MI ROP pen at conceptual stage, where we can have this screening at every NICU level. So it's not something that every innovation can travel to Z. This is another innovation. We are very inch close to Z, which will be commercialized very soon. I'm not sure if you can notice. We learn in cornea, we learn in aqueous humor what happens, the aqueous current. So this is an anterior segment imaging device that we are coming up with very soon in 2 to 3 months, which will show you this kind of high quality images where you can measure, quantify inflammation at a very, very cost effective part. You see the anti-clockwise current, can you see here? All these vessels, all these cells. So journey begins with one step with A, and you definitely leave your footprint for really long if your foot really, really reaches to Z. So thank you so very much for having me here.
Dr. Ashish, and it takes a lot of work to take it till the end, and you have proved it time and again. Excellent work. Any comments, Dr. San? Okay. I now invite Dr. Sayan Basu, who's going to talk to us about 3D printed cornea.
All right, thank you so much for making me a part of this session. And just like Ashish mentioned, I am also not a saint, and I have the following disclosures about the products that I'm going to talk about in my presentation. So the context basically is corneal regeneration, corneal replacement. We are basically interested in restoring vision to patients who are blinded by corneal opacification. In our country, the most common cause is post-infectious and post-traumatic scars. And the problem is that this peculiar indication for corneal transplantation unfortunately does not carry a very good prognosis. Therefore, we have to look at alternative options for addressing the problem of corneal blindness. The other problem that we have in our country, and this is prevalent and true for most of the developing world, is that we do not have enough donor corneas for transplantation even if you wanted to do. So it's a double jeopardy. You don't have enough donor corneas, and even if you have corneas, if you do transplants for patients with inflammatory conditions that lead to corneal scarring, those grafts will not work very long, and then you have to keep repeating those grafts. So what can we come up with as a solution? So this is the problem on your left side. The problem that the solution that we are aiming for is on the right side, which is a biomimetic stromal tissue alternative to replace the scarred corneal stroma with bioengineered corneas, which has the mechanical strength that is required and that the normal cornea has. It has the right curvature and shape, and it can integrate into the host, and just like we suture corneal grafts in place, it should be suturable as well. So that's basically what we are aiming for. So if you look at the cornea, it's a very simple and very elegant tissue, and therefore it is not so difficult to bioengineer it. There are basically three components, the keratocytes and the extracellular matrix, which come together to form the corneal stroma. So if you're looking at a stromal substitute, you have to find a way in which you can replicate the keratocytes, but mostly you're thinking about the extracellular matrix, which is about 90% of the stroma. So 3D bioprinting, what is this? So just, many of you may know how printing works in general, inkjet printers or extrusion printers, that you put in an ink cartridge and there is a nozzle which prints out whatever you want on a piece of paper. You can do this in 3 dimensions, and so this is widely used in industry. But if you use a biological tissue as the ink, instead of printing words on a piece of paper or instead of printing out plastic devices, it can also print out tissue. So that's the concept that we are trying to leverage here. So there have been a lot of bioinks that have been tried in the past. What we are interested is called the decellularized corneal extracellular matrix. We call it the DECM. This is the proprietary product that we have. So essentially, in eye banks, a lot of corneas are wasted because they are not optically suitable for transplantation, essentially because they don't have enough endothelial cells, but they do have normal stroma. We can harness that. Also, when you do a keratoplasty, you throw away the corneoscleral rim, which is not of any use. Now you can also derive the stroma from this. So basically what happens is all the cornea and the corneal parts that are discarded, you can take them and decellularize them, make them into a lyophilized powder, and then use that powder to make this gel, which is called the hydrogel or the DECM gel. We have done a lot of testing with the gel and found that you can apply the gel itself for wound healing, but the gel can also act as a bioink for 3D printing. So this is basically what our workflow is. We are looking, the problem is on the left, the solution is on the right, and so we are looking at different tissue sources. We have now finalized the DECM as a tissue source. Then we are looking at how the different ways in which this can be printed and how we produce the bioink, use the bioink to 3D print corneal tissue, and then we have to do this extensive preclinical testing so that it can finally be then used in patients. So essentially 3D printing, there are 2 basic types of technology that you can use. One is the extrusion printing, which I was talking about earlier. The other is light-based bioprinting, which is more precise and you can print at a higher resolution. This is an extrusion printer in action. You can see on your left you have the printing, and on the right is the printed sheet. So there is a small nozzle from which the bioink is coming out, the hydrogel, and it is printing in 3 dimensions. So you have this sheet of printed tissue that you see on the right side. This is flat, it doesn't have a curvature, but you can use a trephine and then cut it to size, and then you can take this circular piece of tissue and use it for transfer. You can see that it looks exactly like normal corneal stroke. So this is very simple extrusion based printing. You have a printer which prints out a sheet of tissue, and then you can cut that sheet of tissue to size. But what you would like is a little bit more to this, and that you can add a little bit of shape and curvature to it as well. And you can see on the right side that after taking that tissue that we have cut to size, we are now using it in the wet lab to see if it can be sutured as well. And you can see here that the suture is cheese wiring, and therefore this is not suitable for transplantation. So you can also do cross-linking on this, and you can also use molds then to now add curvature and shape to this. So instead of taking a flat sheet of tissue and then cutting it to size, you can actually print a curved tissue using a mold. So here the 3D printer is printing on a pre-defined mold where you have defined the curvature and the shape of the cornea. And then you get a tissue substitute that resembles the cornea more closely in terms of not only transparency, but also in terms of shape and curvature, and you can essentially customize any shape and any curvature using this strategy or this approach. So right now we are doing preclinical trials with this 3D printed customized corneal lenticule. So in the rabbits, basically what we do is we remove some stroma. You can see here that I'm dissecting out a stromal lenticule and creating a defect which is about 300-400 microns deep. And then we use the customized 3D printed lenticules, and we are putting them on the defect that we have created on the rabbit eye and putting them in place. So you can ideally this can be sutureless, and you should be able to suture them as well in case you are addressing a larger defect. Here we are just using fibrin glue to attach this tissue. So this is what basically it looks like. So it requires a little bit of engineering, which is called CAD modeling, to design basically what is the shape and size you want to give to these corneas. And you can also cross-link them to add further strength. So what we do after making these corneas is that we test them in the wet lab to see how they are behaving in terms of handling, that are you able to pick them up, are you able to move them around, and then we will also test them for their mechanical strength in terms of suturability. So here you can see a tissue on the left side which is not cross-linked, and a tissue on the right side which is cross-linked, and they behave slightly better. You can see that the tissue on the right is a slightly firmer, the tissue on the left is a little bit floppy. And then once we have customized these lenticules, then we do the suturing strength to see if they are suturable. This is a term that we have coined. There is no dictionary term which actually spells this out. And what we do is use 10-0 nylon sutures, which we routinely use for keratoplasty, and see when you are tying the knot whether the fiber of the the suture cheese wires or is able to hold it. So here you can see it is able to hold the suture, but the handling is not good. When you're using the forceps, it is a little bit brittle at the edges. So this is not, we are not happy with this. So we want something better. So this is another substitute that we have printed. Here you can see that the suturing is good, the handling is also good, the edges are not brittle and the edges are not cheese wiring. So this is a much better alternative. You can also see that it is transparent. So after having gone through a lot of trial and error and having used different resins also to cross-link. So if you cross-link using different resins, and the tissue becomes a little bit, it gets this yellowish kind of tint. Of course, if you wash it, it goes away. But these are the substitutes that we are working with now. And as you can see the photograph, that they look pretty, they're very transparent and they look very similar to native corneal tissue. And this is the product that we are interested in taking to clinics now. We call this the Neo Cornea. This is developed in collaboration with IIT Hyderabad. As you can see in this video here, I am suturing with 10-0 nylon on a artificial anterior chamber, and you can see that not only is the tissue transparent, but it handles very well. I have absolutely no issues in putting the sutures. Here I've put 4 cardinal sutures, and the sutures are not cheese wiring. It has enough mechanical strength, it has the right curvature, and it has optical clarity as well. So just to conclude, I think the future is very exciting for all anterior segment corneal surgeons, because the number of cutting edge regenerative therapies are on the horizon, including stem cells. We are all assessing their safety and efficacy right now in preclinical studies, but you will have in the future, in the very near future, cell therapy for corneal ocular surface inflammation, just like you have anti-VEGF therapy for neovascularization in retina. And you will have these hydrogels that you can put in with patients with trauma and inflammation that can cause scarless wound healing. And you will also have 3D printed substitutes. And I foresee that this 3D printing will not only be there for corneas, but you will see 3D printing come into vogue in a lot of other applications. For example, contact lenses and also intraocular lens printing. And you can actually customize and 3D print an intraocular lens based on the patient's power and the refractive error. Thank you so much.
Thank you, Dr. Sain. And I think the next session people are already here. And thank you very much, everyone, for attending this.